Hey everybody, so we are talking about DNA. So chapter 16 is all about the molecular basis of inheritance. We started talking in class about the really important scientists whose investigations helped lead to our understanding of DNA. And so what we came up with was the composition of DNA. So we know that there's a deoxyribose sugar, so that's a 5-carbon sugar. We know that there's a phosphate group, and then we know that we've got two different types of nitrogen bases, or two different categories of nitrogen bases, purines and pyrimidines, and then we know that there are two bases that we're looking at for purines and two that we're looking at for pyrimidines. All right, so the DNA backbone, we talked about how DNA sort of looks like if you took a ladder and then twisted it. And so the sides of the ladder, that would be the DNA backbone. And so that is a polymer of sugar phosphate groups. And so what that means is that you're binding sugar to phosphate, sugar to phosphate, sugar to phosphate all the way down the backbone. And there's two, so you've got two sides of a ladder, so you'll have two of those sugar phosphate backbones. Then we have our nitrogenous bases, and so that's what's going to be like the rungs of the ladder. So that's what's bridging those sugar phosphate backbones together. And every time you have a purine, it is going to bind with a pyrimidine. So if you have a purine on this side, it's always going to bind to a pyrimidine on the other side, and each rung is going to be 3 rings across. And so what that means is there's a constant distance between those two backbones, so the backbones are always going to be very straight. And then those nitrogen bases are held together by hydrogen bonds. All right, so here we have our sugar phosphate backbone. So phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. You guys get the pattern there. And now we have our bases over here. So our first one, well, I guess the bottom one is guanine. Okay, and when you look at the nitrogenous base, the sugar and the phosphate all bound together, so base, sugar, phosphate, here you are looking at what we call a DNA nucleotide. So DNA is made up of lots of nucleotides bound together. All right, so this would be like the monomer of the nucleic acid. So you have repeating units of these DNA nucleotides. Here we're looking at another nucleotide. So again, we've got our base. This time we're looking at cytosine, we have our sugar, and then we have our phosphate. Here's another nucleotide. Here we have adenine, then we have our sugar, and then we have our phosphate. Now we have thymine, then our sugar and our phosphate. So this is our last nucleotide up here. This is just 3 different ways of representing DNA. So this one is a little bit easier to see sort of what this overall structure looks like, because you can see the double helix, so essentially taking that ladder and twisting it. Here you have the chemical structure, so a little bit more detail in terms of where things are bonded and how things are bonded. And then you have your space filling model. All right, so we talked in class about all these important scientists. One of them was Chargaff, and we discussed Chargaff's rule. And this essentially was really important for base pairing. And so this is explained by the double helix model, and this rule tells us that every time you have an adenine, it's going to be bound to a thymine. Every time you have a guanine, it'll be bound to a cytosine. And so because you are always binding a purine with a pyrimidine, you're always going to have a 3-ring distance in between the sugar phosphate backbones. All right, so if you look down here, I know the picture is kind of cutting it off a little bit, but you have a purine bound to a pyrimidine, and this is showing you a width that is consistent with the X-ray data. And so you've got 2 rings on this side, a little hydrogen bond to the 1 ring over here. If you were to have a pyrimidine and a pyrimidine, with pyrimidines only having the 1 ring, it ends up being too narrow. So you can see that it doesn't fill out all the space in between the 2 backbones. Whereas if you were to have a purine with a purine, it ends up being too wide. So you can see here that the rings end up going out past where those sugar phosphate backbones are. And so it really makes sense to have a purine and a pyrimidine. The width is consistent with the X-ray data, and this will ensure that the DNA is the same width the entire way. So here we have adenine bound to a thymine. So we have the hydrogen bonding taking place here. And then down here we have the cytosine and the guanine together with the hydrogen bonds. So you can tell that the adenine and thymine need to bind together because there's places to make 2 hydrogen bonds, whereas with cytosine and guanine, you now have 3 places for hydrogen bonding to occur. And so A and T will go together, and C and G will go together. So we want to kind of switch gears a little bit. Watson and Crick published a second paper. This one was a couple years later in 1954. And instead of focusing here on their model of the DNA structure, they focused on a way that DNA could replicate. And then the proof of this replication was then provided later on. And it's really important. I'm going to go back for a second. Because we know that A's will bind with T's and C's with G's, that ties in very closely with how DNA replicates, along with just sort of the overall structure of the molecule. So the replication is the process of making more DNA from DNA. Okay, we know that cells replicate, but we need to understand if a cell is going to replicate, we know that the DNA has to end up in the new cells, in the daughter cells, but how does that happen? How is all of that DNA copied exactly and then passed on to the new daughter cells? Okay, so we've got 3 different models for replication. So these are all possible models for DNA replication. Conservative means that you will have 1 old strand and 1 new strand of DNA. So you'd have 1 double-stranded old strand, 1 double-stranded new strand. Semi-conservative means that you have 2 strands of DNA, and maybe 1/2 of each strand is new and then 1/2 is old. And then dispersive would mean that the strands are just a complete mix of old and new pieces of DNA. So obviously we need to test this, right? And so this is what these models would look like. Okay, so if you had a parent cell, which is this first column, that parent cell is going to have the original strand of DNA. It's shown in that dark blue. Okay, so let's look at the conservative model first. The conservative model says that you're going to keep your old strand and then you're going to produce a new strand. And so you can see here, you've got your old strand, which is that dark blue, and now you've got the 2 dark blue strands together. Here in the first replication, the conservative model, you've got a completely brand new strand, so it's represented in the light blue. Okay, and then when you have a second replication, the same thing is going to happen. This is still going to be your original strand, and now you're going to make another new strand. Okay, with the semi-conservative model, what this is showing is you've got your original strand of DNA. After 1 round of replication, you can see that those blue strands have split apart, and now they are each attached to a new strand, and then the same thing is going to happen in the second replication. With the dispersive model, here's your original strand of DNA. And now you can see that as it replicates, it's showing that the chunks of the original DNA are kind of getting just dispersed randomly. So you don't have one new or 1 old strand. It's just kind of mixed together, and the same thing happens with the second replication as well. So we needed to test this. So we have Meselson and Stahl in the late 1950s. They grew bacteria on 2 different isotopes of nitrogen. So they started growing it on nitrogen 15 and then switched over to nitrogen 14. And so the 15 and 14, those are the different isotopes of nitrogen, so they have different weights. And so what they were able to do is look at the weight of DNA after 1, but then also after 2 rounds of replication. So the experiment, you've got your bacteria that was cultured in a medium that had the heavier nitrogen, so your nitrogen 15. The bacteria was then transferred to a medium that was nitrogen 14. Okay, so then this DNA sample is centrifuged after 20 minutes, so just after 1 replication, and you end up getting your DNA sample. Then the DNA sample was centrifuged after 40 minutes, which was after the second replication. I know you guys may have trouble seeing this. What this is showing you is that the less dense DNA was this light blue band up here, and then more dense DNA went lower into the test tube, and so there was the second band. It's a little bit darker, and it corresponds to the original band of DNA. So it's a little bit more dense. And so what happened was, if you compare these replications, so the first and second replication, to the models that we just looked at, the conservative model, after the first replication, what it would have shown is that you have your really heavy stuff that was all nitrogen 15, and then your lighter stuff, which was all nitrogen 14. And then you would have had again, in your second replication, you would have still had your really heavy stuff as nitrogen 15, and then you would have had more of the lighter stuff up here, because everything is going to just be grown in that nitrogen 14. It'll all be lighter. Okay, this is not what Meselson and Stahl found, so this does not work. Okay, with the semi-conservative model and with the dispersive model, this first test tube actually worked out with like could have been either. So there was just the one band that you would have had with the semi-conservative or with the dispersive model, because you have essentially an equal mix of old and new strands of DNA, so it's a perfect mix of the nitrogen 14 and the nitrogen 15, which is why we see that band in the middle and not like either really heavy or really light. It's a mix of nitrogen 14 and 15, so there would both be in the middle. So the semi-conservative model is in the middle because you have one complete strand of old, one complete strand of new for each of these, and here you have equal parts just mixed randomly. With the dispersive model though, when you kept growing this bacteria in the lighter nitrogen, what would have happened is all of the nitrogen would have become lighter, or I'm sorry, all of the DNA would have become lighter, because how it's all growing there, and that old nitrogen, the heavier nitrogen, is just getting mixed up randomly. What we actually found though, this was not correct. What was actually seen though was that you still had your sort of medium band, and then you had a lighter band, and so that corresponded to the semi-conservative model, where you still have the old strand of DNA mixed with the new, and then as it replicates in that nitrogen 14, you're going to have your newer strands that are all a little bit lighter. So the results confirm the semi-conservative model of DNA replication. And so now we kind of understand that it's a semi-conservative, so that you're always going to maintain like an old strand and then pair it up with the bases to make the new strand. And so there's three like really basic steps that happen in DNA replication, and then we will get into the details later on. But first, the origin of replication. In order to replicate DNA, you need the DNA parent molecules to split apart and break those hydrogen bonds between the nitrogenous bases. So we are talking about like the rungs of the ladder. You need all those rungs to split apart in order for DNA to replicate. Once that has happened, then DNA is going to build the missing backbone using the bases on the old backbone as a template. So what that means is you're going to have enzymes coming in, and they're going to be looking at the old template. And so if this is an adenine on the old template, then the enzyme is going to bring in a thymine to pair with it. And the same would work if you have a guanine, then the enzymes would bring in a cytosine to pair with it. So during elongation, enzymes are going to use that old DNA backbone as a template to create the new side of the DNA molecule. After that happens, then you're going to have DNA repair. And so this is when the new DNA strands are proofread at the end of replication. So obviously keeping the DNA molecule correct is really, really important. And so the DNA is going to have to be proofread after the replication takes place so that any mistakes can be fixed. So this is just, again, showing you, so here's your parent molecule. You have the strands separate, and now you have your daughter DNA molecules. And so because we had a guanine here, an enzyme brought in a cytosine here. Because we had an adenine here, an enzyme brought in a thymine here. And again, you can see that it is semi-conservative. So the dark blue strands are conserved. These are the original strands, and then you are going to build new strands to complement that. All right, so the lecture question. I just want you guys to kind of summarize in your own words, how does the structure of DNA allow for easy replication? So again, shoot me an email with your response, or actually respond on the class website. That would be even better. All right, guys, thanks.